So, I will discuss multi-scale models of basal ganglia in learning and conscious processing. I didn't give this title, someone gave it to me, maybe it was Auser or something. So I will try to make an effort also with this conscious processing. But since this is sort of a cortical meeting that bridges over to consciousness, I'm not aware that everyone knows so much about the basal ganglia, so just some basic facts first. So the basal ganglia is sitting in the forebrain. This blue and green thing is the input stage. It looks like they are separated, but actually they are also connected. It's just the internal capsule that divides them a bit. And then we have this red thing, medial to that is the globus pallidus that consists of an external and an internal part, and this is the output stage of the basal ganglia. So these are telencephalic structures. They are sitting sort of below and lateral to the lateral ventricles. Then another nuclei in the diencephalon subthalamic nucleus is also counted to the basal ganglia system. And we have another output nucleus, which is very similar sort of in function and projection as the output stage of the globus pallidus, and this one is called substantia nigra and sitting in the midbrain. And there we also have substantia nigra compacta, which is dopamine neurons. So the basal ganglia receives input then from all these other telencephalic structures, such as the neocortex, the limbic cortex, hippocampus, and amygdala, and so on. And these inputs from the telencephalon, it goes to the stratum and then to the output stages of the basal ganglia, and then it goes downstream to control behavior, but it also goes back to cortex or back to stratum in these loops. And these loops are closed, but there are also open loops. And so that is in general true, but maybe I should also highlight that if you go into the striatal tissue and look carefully, then you would see these so-called patches or striosomes that don't really follow this scheme. They receive input from the limbic system, and then they control neuromodulatory systems in the brainstem, like dopamine and 5-HT. So then what about function? This is now a more mouseified illustration. So we said we had two output stages that are quite functionally similar to each other, globus pallidus interna and substantia nigra reticulata. And those are tonically active and inhibitory, and they keep then centers in the thalamus and in the brainstem under tonic inhibition. And this inhibition can be increased or decreased to evoke behavior. And this is done in a push-pulled manner by so-called pathways that goes through the basal ganglia system. And the most well-studied are the direct pathway that provides sort of a go signal, start movements or gates through this loop back to cortex, and the indirect pathway that provides sort of a no-go signal. And this organization is very much evolutionary concerned, when even down to the lamprey that has been studied a lot in Stockholm. We have the same cell types, sort of, and these different populations, they connect to each other as in higher vertebrates, even though, of course, the macroanatomy is very, very different. So this suggests that the function is sort of quite conserved or generic. And the function has been said to be decision-making, or if you think about motor behavior, action selection. And this has been phrased recently by Arber and Kostel, like if the cortex broadcasts the wish, the basal ganglia will decide whether the wish should be fulfilled or not. And exactly what wish it is, it depends on what part of the cortex that sort of broadcasts this wish to the basal ganglia. And these decisions are then adjusted due to reward learning. If something goes wrong, you can get like motor disorders if dopamine goes wrong, like in Parkinson's disease, or various neuropsychiatric disorders that also typically involve the frontal lobe. So you have the frontal cortex and basal ganglia thalamic loop, so working memory, attention disorders, ADHD, schizophrenia, and so on. So obviously, basal ganglia is important for attention and attentional switch and behavioral flexibility. And in line with that, it also regulates neuromodulation, which is also important for this. But what about consciousness? That was a tricky part, because usually you don't talk about basal ganglia when discussing consciousness, sort of as for a hippocampus. But basal ganglia is known to be important for the attentional blink, for instance, and maybe we will hear later today that it also plays a role when it comes to this perception threshold in the somatosensory cortex. And I also found a quite recent publication where they had recorded from the monkey brains and found that in the stratum, in the parietal cortex, and in the thalamus, you had the highest sort of estimated integration of information. So if you believe in this IIT model, then for sure basal ganglia should play a role there. But usually the basal ganglia can actually, it's involved in procedural learning. So actually it can take something which has been conscious, and then it can perform it more in a habit or implementing intuition. So for instance, there are studies from these Japanese chess players, or I think it's called shogi, that game in Japan, where they see that when you get a skilled shogi player, then it's a stratum that is becoming active, but in the beginning when you try to think to play the game, then it's the parietal cortex. And also I think it was a good example what Ausra said about this person, what was his name, Clive Wearing, that he couldn't remember, but he could still play. That was definitely involving the basal ganglia loop. So now I will just, I will just sort of ask and think that what part is really necessary for consciousness. And then I will show some old work from Sweden that I used to show to students, because it's a bit interesting. So at that time, in the 70s, they removed neocortex from birth bilaterally in kittens. And these kittens could, and this has also been done in mouse and rodents and rabbits, I think. But these kittens could live in lab environment for years. They could even have kittens that they sort of fed at least. And this is a illustration of when such a cat tries to step down from a box. These cats can get hungry or thirsty, search for food, remember the location of the food in a tea maze, find the way out of a maze, and they don't really socially interact with other cats, or the other cats didn't like them, but they could sometimes display aggressive behavior. So a large part of the neuromachinery for goal-directed motor behavior is obviously subcortical. And now this is just a short movie of an intact cat and a cat without cortex. So this is an intact cat that they put into this maze. And probably this cat is conscious, at least. And now it's an example of a cat that didn't have any neocortex from birth. So here it comes, and the first observation is that it's not particularly handicapped, even though it lacks the motor cortex and the pre-motor cortex and so on. Finds a way to the next room. And then it looks like it is fooled by this black square in the wall. At least it looks like that. Which is quite interesting because it lacks a visual cortex, so it's blind, but it has a thalamus left and eye, so it probably has some rudimentary feeling for maybe dark or light or whatever. So is this cat conscious? And I think we should have this question in mind for the rest of the symposium. did this cat experience anything at all? And I mean, to me, it seems like it did. It could be angry, it could get hungry and so on, but who knows? Now I will be more concrete and go back to the basal ganglia. So I said that the basal ganglia, sort of in a push-pull manner, gates through information going back to cortex and going downstream. And that is heavily neuromodulated by dopamine, which can, to a first approximation, be explained by this direct and indirect pathway. So if you increase dopamine, then you facilitate sort of this go command through the direct pathway, because these principal neurons, the striatal projection neurons, carry dopamine type 1 receptors, which is then sort of depolarizing these cells. But the indirect pathways, striatal projection neurons, they carry D2 receptors, so this pathway is a bit inhibited. So if you increase dopamine, it's sort of more of a go command. And the opposite is true if you decrease dopamine. So how then can you understand that dopamine is a signal important for facilitating a behavior? Then dopamine and also actually acetylcholine are transiently increased just before the onset of movements, and this is sort of a conceptual explanation of that. If cortex is silent, striatum is silent. If cortex is active, striatum sort of sense that, but it has to have, in addition, dopamine around to really react on that and gate through the command according to this idea. Then one should also, so this is one of the things that dopamine is doing in the basal ganglia, but also dopamine is a reward prediction error. It is involved in learning. And there we have some other sort of features, that first the action must come and then the reward, otherwise you don't learn. You cannot have the reward and the action and so on. So there dopamine just comes after, while here it comes first. So what is deciding then how to interpret these dopamine signals? When should learning occur and when should sort of facilitation of excitability and so on occur? So obviously we need to have, and this kind of information processing is taking part at the sort of the receptor level and in receptor-induced cascades. So I also just wanted to start at the systems level and just illustrate that we can also today, as for the hippocampus and actually somatosensory cortex and motor cortex and the cerebellum and the visual system, build these systems in a very detailed manner, sort of bottom up. So when you build this system, what you need is type of neurons, morphologies, expression of ion channels, electrophysiological properties. You need to know how many cells are there, connectivity and synapse properties. And then, of course, the workflow is first of all you have to have critical data, and then you can build a sort of a database of detailed neurons, and then you put them in the volume you want to simulate, connect them, and then you can constrain synaptic properties and do simulation experiments. So we start with this first step, just to show that when it comes to the striatum at least, and also for the rest of the basal ganglia, we know cell type composition in the striatum. 5% interneurons, the other things are constituted by these striatal projection neurons. Most of the interneurons are inhibitory. We have the whole transcriptomics, so we know which cell types can express which ion channels. So when we have morphologies, we can then start with putting these models of these ion channels distributed on the cell surface to see if we can reproduce these experiments using this optimization workflow that Michele discussed. And here, of course, it's really critical that we have all these public repositories when it comes to data and tools. And in this way, we can generate models for approximately 98% of the striatal neurons. There are a few exotic ones where too little electrophysiology data is there, and also this is ongoing for the rest of the basal ganglia. And then it's time to build the network. So then, of course, you need to know the structure. If you want to model, for instance, the whole striatum, you need to know the volume and the boundaries and so on. And you also need to know the density of cells, and maybe if there is some gradients over the tissue in different parts of the striatum. And you need to know the connectivity and how the connectivity decays over distance. All this type of structure information. And then to set up the network, we use a tool called snudda, which means touch in Sweden. So this is an illustration. If this is a stratum and you want to, you look into a part of that, then you can place cells with the right density. And if you have also dendrites and axons, it looks quite crowded, and you can do touch detection where axons and dendrites come close to predict where connections could be, and then we apply pruning rules, which is partly inspired by Cortix work. And these pruning algorithms contain different steps. You can detect all the contacts, remove a fraction of synapses, do distance-dependent pruning, make some sanity check, not too few, not too many. Also remove fraction of connected pairs. And then when you have done these steps, you come up with a sort of a final prediction of for particular pre- and postsynaptic neuron types, resulting pairwise connection probability with distance. And you know the number of connection sites per coupled cell pair and number of pre- and postsynaptic neurons. And these kind of predictions you can then compare to what is known or estimated based on data. Yeah, and also, of course, one needs, very importantly, short-term plasticity. And I should say that most of this data actually comes from the Karolinska Institute and Silverberg's lab there. And then it is, so these bits and pieces you can then put together in these automatized modeling pipelines. So for instance, this is from a previous publication where we can model a piece of the stratum and look at what happens, for instance, if check that it looks reasonable when you get a cortical command or a dopamine command and so on. And these in silico platforms can then be used to perform in silico experiments that are hard to do in vivo or in vitro. For instance, some lesion experiments to better understand the role of different neuron types and how neuromodulation affects this. And what I think is also very interesting, we try to illustrate that, is that you can use these biophysically detailed models to test hypotheses from more phenomenological models and see how, when it comes to mechanism, is it so that the biological tissue can actually do these things that have been suggested. So that is a challenge, I think, to bridge the gap. Yeah, and we can investigate how, for instance, synaptic clustering could affect the network and so on. So there's a live paper of that on this first platform paper on eBrains. Yeah, this is just an example of what I mean with experiments that perhaps are not so easy to do in experiments. So you can maybe do some selected lesion experiments in the connectivity and look at what happens if you remove particular inputs on the spike frequency of these neurons when you give a cortical command, or perhaps when you have dopamine. So we have dopamine and acetylcholine effects also implemented, pre- and postsynaptic effects implemented in this network. And one can also ask what are the consequences for plateau potentials and synaptic calcium that I will come back to. So, I mean, obviously it's a very good tool if you want to do in silico experiments. So now I will switch sort of to the cellular level, and it's obvious that when you build these microcircuits, you also get detailed models of single cell data. And over 10 years ago, it was shown that also in the straight principal neurons, you have these plateau potentials. So we wanted to simulate that. What happens if we have clustered inputs distally or proximally? This is just an example when you activate the cell with proximal clustered inputs, then the cell depolarizes and then this depolarization dies out. But if you do it at the distal dendrites, then there is a plateau potential and this depolarization stays on for some time. And during this time, obviously, we have facilitation of spatial and temporal integration of excitatory inputs. This is expected. And also it's known from other systems that plateaus facilitate synaptic plasticity. That was mentioned, for instance, by Michele in the hippocampus. And this is also true for cortex. So what we wanted to understand better is how these plateau potentials could be controlled and shaped by the local network, because most of the neurons in the stratum are inhibitory. So then we can simulate what happens if we evoke a plateau in the distal dendrite. A plateau is sort of the same as an NMDA spike. And have proximally GABAergic input or more distal one, and so on. And to make a long story short, we can say that proximal inhibition is typically most efficient to prevent spiking if you activate the neurons using distributed inputs or using current injection in the soma. But it doesn't help much if you have a plateau potential evoked in the distal dendrites. Then you need to have distal inhibition close to, or in that branch where you evoke this plateau potential, and this could then potentially also affect plasticity. So this was also actually verified in experiments together with collaborators, that you can sort of kill a plateau if you have distal inputs, but not if you have proximal inputs. And this has also been shown in models in cortex by Idan Segev's group. So what I said so far is that it's possible to reconstruct the striatal microcircuit and also the rest of the basal ganglia with the data that is out there. And this data is actually accumulating very rapidly. We tested this cell type specific inhibition. And if this is true, which we believe, then it means that only a few distal GABA synapses can actually influence the cell-wide spatial temporal integration and plasticity, even though when you do this pairwise recording, you hardly notice any effect. So I think this is quite interesting. And these things we also use, one can also use that to investigate how nonlinear patterns can be learned due to dendritic plateau potentials. And I just wanted to show a bit of this. This was something that we discussed a lot with Walter Sen. I hope we can continue that. So this is a sort of well-known feature binding problem, and there are different solutions to this. But one thing is you can capitalize on these things with nonlinear dendrites, and this is just an example. The feature color and shape. So maybe you should recognize a red strawberry and a yellow banana, but not a yellow strawberry or a red banana. And this one way to implement this is you assume that this evoke a plateau potential in one dendrite in the neuron, and this evoke a plateau potential in another place of the neuron, so that these neurons might be spiking more easily. But if you give the wrong combination, then the neurons are silent. So this is sort of a conceptual illustration of that before learning it might be like that, then it can't distinguish, and afterwards it has learned to distinguish. And I mean, this has been discussed in various publications, and we have people in the audience that have modeled this. And what we wanted to know is if we could reproduce some of the things actually that Walter Sen had in a phenomenological model before, but just build on what is known of the mechanism in this straight up projection neurons, using only local synaptic cues, nothing else. So there is known, this is a plasticity mechanism, and it's known that LTP needs NMDA calcium and dopamine, LTD needs L-type calcium and no increase in dopamine. And these things we can simulate, we can plot L-type calcium, NMDA calcium, and we can also see how many inputs are needed if we assume that the inputs, the synaptic strengths have undergone LTP or LTD and so on. So then we sort of implement that sort of plasticity rule, and there we notice that homeostasis mechanisms are very important, but I will not go into that now, but that's quite interesting insights that makes it robust. So then we can start with, for instance, that situation, some background noise, and then sort of randomly distributed combination of these features, and then after training, you can then, for instance, have a plateau potential when you show the yellow banana but not the red one, and then it also likelihood is increased that it spikes in the soma. And this can be done then in population of these neurons. So after training, it means that if you show the wrong combination, the neuron is silent, but if you show the right one, it is spiking. And this is interesting that when you randomly distribute, then some neurons can learn this nonlinear feature binding problem, but some just pick up one of those and not the others, especially if you start with these conflicting things where you have to do some symmetry breaking, then of course it doesn't work. But this is when you train every neuron individual, and this doesn't work in the brain, I guess. You have to do it population-wise. So when you then rerun these simulations, but just take a majority vote and train the whole population, then it still works, which means that you have subthreshold learning, and at least in the stratum it's known that you don't have to have spikes to have plasticity. Obviously, it's well known that you have to have enough nonlinear plateau properties, otherwise you cannot do this, computing. Otherwise, yeah, it cannot distinguish that. But then we have also played around with GABA plasticity, which actually I think is quite interesting. Our GABA plasticity is just explorative, but I really would like the experimentalists to focus on GABA plasticity as much as glutamate plasticity. So I will not show any results, just say that we and others have also, of course, modeled these receptor-induced cascades, underlying phenomena such as this timing dependency in reward learning. Why is it so that there is a delay required? And something else we have been interested in is to understand better using models how these transient neuromodulators can be detected. And I should just say that it's really difficult to constrain model parameters at this level, and within the Human Brain Project we have also worked together with molecular level simulation labs that can predict parameters for these kind of models, and there are live papers on that and also. So I think this is sort of an interesting future that will of course continue. Nothing would have been possible without the eBrain tools and workflows and open source data repositories that have accumulated over the years. And also nothing would have been possible without all these talented and creative people that have helped to build this straightal microcircuit. I should also say that Sarah and Daniel was the one who did this learning in these nonlinear neurons. And also we have many other international and national collaborators like Maria-Lena and Hauser and so on, which also are very valuable, and the funding is really necessary, otherwise these people would not have worked if they hadn't got salaries. So thanks for that.